come from the world-famous school of Otto Creutzfeldt's School of Neuroscience in Germany. And Wolf has made absolutely seminal and game-changing discoveries in neuroscience, in neurophysiology. I don't really want to enlist the fantastic discoveries of Wolf, but of course, if you have ever heard about oscillations in the brain, and there is, of course, a long German tradition von Malzburg and others who are behind it. But the most significant figure is definitely Wolf-Singer, who realized for the first time, and not only realized, but has demonstrated crystal clearly that oscillations of far away neuronal populations, assemblies, synchronous oscillations, play an absolutely key role in the brain. And I hope that tomorrow we will hear about it from Yuri Buzsáky also. But just one more comment about Wolf. He is not only a neuroscientist, he is really a humanist who is so keenly interested in, let's say, the wider applications or interpretations of neuroscience. He is regularly in India, participating in different activities, including meditation courses. And yes, Wolf, you told it a number of times to us. Well, I hope that he will, in his talk, give a larger picture of neuroscience, which is very relevant to our present-day conference.
So, thank you for attending. I just have to correct this word. I did a crash course in Zen meditation for a fortnight, but that was it. I know what it is, but I'm not a regular meditator. I think in the talk, and I hope it will become clear what we actually do not know. I think we are still at the very beginning of understanding the complex workings in the brain, and I will give you some aspects, hoping to be able to demonstrate how little we know about essential features of this mysterious organ. Relations matter. I think this is obvious and trivial and very nicely illustrated by the concert of Frederick Uhl yesterday. He used the same tunes in order to play Bach, Chopin, and Ligeti. So all that matters is the way in which they are recombined, the relations among the tunes. The same is true for the genes. We share most of them with Drosophila, but we look very different because the way in which they are orchestrated is so much different. So this, of course, applies for all objects of the world that surrounds us. Therefore, cognitive systems need very efficient mechanisms to detect, classify, and encode relations. And this is what I will be talking about mostly. Now, a common strategy in nervous systems to encode relations is convergence of feed-forward connections. Neuron A representing one feature and neuron B representing another converge onto neuron C. If C is thresholded appropriately and fires only if A and B are the case, then neuron C is a conjunction-specific neuron expressing with its responses a particular relation. There is an alternative view proposed by Hebb in particular, saying that if a bunch of neurons representing different items and contents are linked together by temporal coherence, by firing together, they as a whole express the relatedness of the features that are represented by the elements. So, ultimately it comes down to the fact that all relations are defined by temporal contiguity, because even in the feed-forward recombination scheme, what matters is that the inputs arrive simultaneously at the conjunction-specific neuron C. There's also more or less circular relation between the learning mechanisms that are implemented in the brain and the way in which we experience relations. We all infer meaning from temporal contiguities. If something happens simultaneously, we tend to associate these events. If one event happens before the other, we tend to assume a causal relation if there is some consistency. Pavlovian conditioning is an example. Now, it's interesting that all learning mechanisms rely on the evaluation of temporal contingencies, whether it's short-term spike time-dependent plasticity or other forms of Hebbian plasticity. They all evaluate the contingency between pre- and postsynaptic activity in order to change the gain of synapses. So this means that all meaningful relations that are generated in the brain need to be expressed or encoded into lasting connectivity changes. They need to be recoded somehow in the temporal domain in order to satisfy the requirements of the learning rules. Be it spatial relations or temporal relations or symbolic relations, at the end what matters is that things come together at the neuron at the same time, or with some time delays. So the feed-forward strategy to evaluate and detect evaluations is implemented in most nervous systems and is probably the only mechanism in simple nervous systems of insects and in certain basic systems like the olfactory system in higher vertebrates. It's called the labeled line code because after convergence you have a conjunction-sensitive neuron that stands with its responses for a particular relation. And this is also the strategy that is implemented in most AI systems, in particular in the now very popular deep learning networks. Here's an example. You have an input layer with feature detectors, an array, very large arrays that extract certain features from patterns. Their signals are then redistributed onto a second layer, recombined by divergence and convergence, and this process is reiterated several times, therefore these networks are called deep networks. Now, what is done in AI is one presents a large sample of different patterns at the input, a large sample of patterns at the output, and then using a backpropagation learning algorithm which is not physiological but essentially mimicking a Hebbian mechanism, one tunes all those synaptic weights at all these connections in all those layers sequentially, step by step, until a particular input pattern produces an output response of a classifier unit that is stronger than all the others. This is how these deep learning networks function. They are purely feed-forward, and they use a very unphysiological synaptic plasticity mechanism. Now, as you already heard, there are a lot of new data on connectivity and dynamics from neurobiology that call for an extension of this rather simplistic view. We now know that the density of interconnections is unexpectedly high at all scales. Does this work? Yeah. No? Not very good. Is that better? Good. According to Henri Kennedy, about 70% of all possible connections among the nodes being cortical areas or columns, are actually realized in the brain. This is much more than you would actually expect from a small world network. Then we are very much impressed by the prevalence of reciprocal connections. A protects to B, B protects back to A. This is both true within layers as well as across layers. Usually the feedback connections are 10 times more than the feed-forward connections. So there's massive recurrency in this system. And there's a soft hierarchy, as you will see. And since the times where we started to record from more than one place in the brain simultaneously, from more than one unit, we realized that there's an extremely complex dynamics. There are higher order correlations, there's high dimensionality that, at least for me, goes beyond what I can imagine. It's just mind-boggling complicated. So here's the one example of a connectivity graph of the primate, essentially visual system. These red areas back here, the red dots stand for cortical areas, mainly concerned with vision, and then a few selected ones in the prefrontal and frontal cortex that deal with the control of attention. And these white fibers, they are connections, reciprocal connections among all those areas. And you see that from primary visual cortex onwards, where the feed-forward principle is still visible, even though all these connections are also reciprocal, there is a dense meshwork of connections that interlink areas with one another. And if one knows one's way in this complex scheme, one can get from any neuron to any other neuron in this network with a maximum of 1 or 2 switching stations. So this is quite remarkable. And the same connectivity scheme must hold in the human brain as we see here with TTI pictures. So I would like to take you through a brief Gedanken experiment. This is the sensory cortical system of a cat with a few visual areas. It has much less areas than the monkey and even much less than humans, of course. Just have heard there's about 180 in humans altogether. So visual areas, tactile areas of the somatosensory system, auditory areas, and then here the limbic areas whose function is to associate emotional connotations, value, to whatever these areas have processed and presented as a result. Now imagine, and these fibers here are anatomically confirmed massive reciprocal connections among those nodes in the network, cortical areas. Now imagine that this system is confronted with a polymodal object, like you have this system, you look at a barking dog and touch its fur. And there may be in the scene also, in addition, a cat. Let's start with the dog first. In this case, a very, very large number of neurons distributed across these visual cortical areas will become active, namely those neurons that respond to features that are part of the visible aspect of this dog. The same will be with the neurons in the auditory areas that respond selectively to certain features of this barking noise. And the same will be true for the neurons in the tactile system if you touch the fur of the dog. And at the same time, the limbic system will find out whether the barking and the posture of the dog is aggressive and whether you should be afraid or not. Now, the question is, where is this dog represented in this network? And the answer is, one could not point to any particular site in this network. The representation, which is not further reducible of this very concrete polymodal sensory object, is a widely distributed spatiotemporal cloud of activity that needs to be structured in some way. Now imagine that at the same time there is the cat, and you see the two in relation to each other, then also the cat needs to be represented here somehow by a cloud of activity. And this raises a number of interesting problems. It's a very versatile way of representing things because the same neuron can at one time, depending on the constellation, participate in the representation of a dog, in the representation of another object, and so forth. So it's a combinatorial code. But it comes with a lot of problems. The question is, how are the various features of these objects related to each other? How is it made sure that neurons coding for the features of the same cognitive object, the dog versus the cat, are in any one moment in time identified as members of a functionally coherent ensemble? And then another big problem in such a distributed network is, how is it assured that communication be restricted to selected senders and receivers? It is absolutely inconceivable that what a neuron says is distributed widely through all its connectivity to all the others. That would be similar to tuning your receiver to all stations, radio stations in the world. It would just end up as white noise. So there needs to be selectivity in the routing of activity, and that needs to be regulated in a context and goal-dependent way. I think this is obvious. Just an intuitive example of the problem, if you have to segment the scene, you have to make sure that your visual system associates this white surface with this black surface, but this black surface not with this black surface, even though they are similar in contrast, in order to end up finally with the percept of a horse. And if I gave you enough time, you would find 7 of them. I don't give it you. And this just illustrates that there must be rules that allow the visual system to do this sorting of figure elements and ground elements and bind the right elements together. These are Gestalt rules. They are wired into the system to some extent. They are learned. So I had said in the beginning that if we want to encode relations and imprint them into circuitry, one has to convert them into temporal relations, because this is what the learning rules understand. They don't understand any other signature of relatedness. So if this were so, this would necessitate that activity needs to be somehow structured temporally. It's no problem if stimuli from outside imprint temporal structure on neuronal activity, like the response from the tail sensors of Aplysia and a response from another part of the animal, that need to be associated. If the stimuli are simultaneous, the activity will arrive simultaneously. But there are many things that are encoded not by transients, but by sustained activity. So if they need to be bound together selectively, this activity needs to be structured temporally somehow. So in addition, in order to associate neuron groups dynamically in a goal and task-dependent way in this large network, in other words, in order to form functional networks, distributed activity needs to be temporally coordinated on the fixed backbone of the connectome, because the synaptic gains cannot be changed on the fly, but the networks need to be restructured on the fly depending on the task. So if we had a cue for this, or a solution to this problem, it would probably be a rather general solution. And there is some supportive evidence for temporal structure in self-generated neuronal activity, in addition to the structure imposed by the environmental transients. It is that most microcircuits have properties of relaxation oscillators. This is because of the feedback loops of excitation inhibition. So virtually all local microcircuits can, in principle, oscillate, and most of them do. Now, there's a theorem, if oscillators are reciprocally coupled, and much of what we have seen is reciprocal coupling, they can synchronize according to a formalism that is called the Arnold tongue. The closer their genuine resonance frequencies, the easier they will be entrained in synchrony. If the preferred frequencies of these local oscillators differ too much or differ more, then one needs stronger connections in order to synchronize them. And if they have two different preferred frequencies, they will not be able to synchronize. To entrain oscillators into synchrony is a very energy efficient strategy. As you probably know, the experience of Van Huygens, it was a Dutch watchmaker who one morning came into his workshop and saw that all the pendulum clocks that he had were either swinging in synchrony or in counterphase. He was very puzzled by it until he found out that he had hung them on the same timber. And the small vibrations of the pendula of these clocks propagated across the timber down into the clocks, and over the night had synchronized them either in phase or in antiphase. So synchronizing oscillators is something you can do very easily without much energy, and it can be very fast. 1 or 2 cycles may be enough. So what we see is we see oscillations in different frequency bands, and I think Giri will talk a lot about it this afternoon or tomorrow, forgot. They range from 0.1 hertz all the way up to 200 or more. Then we see that these oscillations can synchronize, they can show phase shifts, consistent phase relations, or they can shift. And we also see an interesting phenomenon, cross-frequency coupling. We have low-frequency oscillations. It was mentioned yesterday, the theta rhythm in the hippocampus modulates the amplitude of a superimposed gamma rhythm so that in a particular phase of the theta rhythm, the gamma rhythm increases and then decreases again. This is an interesting thing of cross-frequency relations, which may have some importance in expressing message relations, but this is still a hypothesis. So what do we see? And apologies to those who are neuroscientists, you will be bored by it, but if one records from a bunch of neurons, in this case in the visual cortex, activates them appropriately, they have a high tendency to engage in this burst firing, and you see these are clusters of discharges, and the envelope of this activity, the so-called field potential, shows a fairly nicely regular oscillation. And this signal is now used very much in order to identify functional networks by measuring the coherence between these oscillations. And this type of oscillation is found in most of the structures that have been investigated. Frequency ranges differ. I will talk mainly about the gamma frequency range, which is 40 hertz, mainly a local phenomenon. So what makes these oscillations interesting? If one records from 2 spatially segregated sites, 2 different clusters of neurons, that can be activated because they have superimposed receptive fields, this again visual cortex, by a coherent object. Neurons discharge and they engage in this oscillatory firing, and the cross correlation shows that these oscillations are precisely synchronized, you see from this modulated cross correlogram. Fine. This is interesting, but it could just be a consequence of these neurons being strongly connected. But it is more interesting because this synchrony comes and goes in a context-dependent way. If one activates those two classes of neurons with two segregated receptive fields in visual space, with a coherent stimulus, a continuous bar, which is one object, then they synchronize very nicely. If one activates those two neurons with two bar segments, this is not shown here, that move coherently with the same speed in the same direction, so it would look like this, they would also synchronize, even though not as strongly. But if one activates the same classes of neurons with two bars that move in counterphase, which is certainly interpreted according to Gestalt principles as two different objects, they still respond, as you see with the black surface here is the number of spikes, is about the same as here. But they're no longer synchronized. So synchrony tells you something about the coherence of the stimuli, about the relation between those activating stimuli. The mechanism is close to trivial. If a neuron engages in an oscillatory process, this is because it gets pulsed inhibition at a particular rhythm because of the pacemaker function of the inhibitory network. So a neuron goes from a phase where it is depolarized, highly excitable, can emit spikes and receive EPSPs and convert them into spikes, into a phase where it becomes less and less responsive because there's a strong inhibitory volley that shunts the membrane and hyperpolarizes it. So in this phase, the neuron cannot fire, and if EPSPs arrive, they will not drive an action potential. That's all. If two neurons that are interconnected happen, for some reason, to oscillate with the same frequency in the same phase, you see that they can communicate, because when this one sends, this one is receptive and vice versa. This is also the condition where synaptic plasticity can occur, because here the probability is high that EPSPs converging with postsynaptic activity in a very depolarized dendritic segment will activate NMDA receptors, produce calcium entry, produce all the cascade that leads to synaptic gain changes. Nothing of this will happen if another neuron group that is also connected as efficiently as this one with respect to anatomical connection oscillates in counterphase. In this case, signals from here arrive when this one is refractory and vice versa. So there will be no communication between those neurons and there will be no synaptic plasticity. This is the mechanism that converts synchronous or asynchronous activity into lasting changes of connectivity and can be used to associate assemblies and express relations. So I don't go through the evidence because most of this is published by other lab, by my lab, a lot by other labs. There's also still a controversy, mind you, people who don't think that this is at all a mechanism that is reasonable. But there's evidence that this mechanism is used for feature binding, particularly during development of neuronal architectures. There's good evidence from the lab of Bob Desimon and others that it is used to select signals through attention, because if you synchronize neuronal discharges, they will be much more effective in propagating forward because synchronous volleys are triggering action potentials in following neurons more easily. And you can open communication channels by synchronizing sender and receiver to the same frequency. It's, well, this is slightly circular. Measuring this coherence or this synchrony is used for the diagnosis of functional networks. So people think that functional networks are defined by coherent activity. And there's more and more evidence that disturbance of these synchronization patterns are associated with pathological processes, and much work has been investigated in schizophrenia and in autism research. And if I have time, give you some indication that and Stanislas may probably talk about it because he did a lot of work on this, is establishing a close relation between the massive binding that needs to occur during conscious processing and the occurrence of large-scale synchrony. So I give you only one example of disturbed dynamics in schizophrenia. Normal subjects and healthy subjects and patients with the diagnosis of schizophrenia, which I think is a very heterogeneous diagnosis, but this is what the clinicians told us, had to find out whether these Mooney figures were faces or not. So a simple cognitive task just to keep them busy. Then we recorded MEG, made a time frequency analysis of the wave patterns after source localization with the beamformer, and then investigated both the power of oscillations in different frequency ranges and, most importantly, the precision with which oscillatory activity in the gamma and beta frequency range was synchronized across larger distances of the cortical network. And what you see in normal subjects is that at a particular moment in time after presentation of these Mooney faces, when they engage in the recognition process, in a particular frequency range, plotted here on the ordinate, 40 hertz and slightly below, so this is the high beta, low gamma range, there is a moment of very high phase synchrony that is very precise, that spreads over large cortical domains, which is much less expressed in schizophrenic patients, population comparisons, same thresholds. And there's a fairly good correlation with the symptoms given to us by the clinicians and this decrease in phase synchrony. And one can then use phase synchrony in order to map those functional networks. And in normal subjects, you see that even this simple task engages a large number of different cortical sites, and all these black things are showing sites that show this strong phase synchrony in this range of latencies. And in schizophrenic patients at the same threshold level, there's much less of this. So we see something very similar in autism spectrum disorder, and most of this has been confirmed by other labs since. It could be a cause of the dissociative symptoms because it could be responsible for false binding operations and for the inability to bind together what should be bound together. Whether it's a viable endophenotype will have to be seen. Now, I now want to switch gears a little bit and talk about functions that are not accounted for by conventional computational strategies, the strategies that I have been suggesting so far. And this is, first, the ultra-fast access to a vast amount of stored memories. You can immediately say if you know something or not, within a fraction of a second. Also, you need this fast recall of stored memories. Then you have to compare sensory evidence, sensory signals, with stored priors, with stored knowledge, because you need to do this every time you shift your gaze, and you do this 4 times a second. You get a new input pattern from the retina. You have to pull out from your store the right template, the right a priori knowledge that you need in order to interpret the sensory signals that are sparse and unordered. This is required for predictive coding. It means every 200 milliseconds you have access, you can retrieve from this immense store of experience the right template that you need in order to interpret the incoming signal. Sometimes it takes a bit longer, but usually it can be done very fast. This means that all these priors must be stored somewhere in a huge storage space where they can be accessed very, very quickly. And the question is, how can this be done? And before, I would like to give you a brief example that shows how important it is to recall, to rely on stored knowledge in order to interpret what you see, and that actually what you see is not what's out there, but is what this matching operation has found out. So here, many of you may know this example. I claim that A and B have exactly the same brightness. You doubt that. I can prove to you that it is really true, just by taking away a little bit of the context. So, they are really have the same brightness, so the same amount of photons is coming from here as from there. But it doesn't look like it. So why doesn't it look like it? Because the brain as a whole, the visual system, also sees this column here and the shadow thrown by this column. And then it starts to calculate and says, if the same amount of photons come from here than from there, but there is a shadow, this must be brighter. And then it computes it bright, and what you see is the result of a computation that you are not aware of, which is a result of a Bayesian matching operation. So, the hypothesis is that the storage of the vast number of priors, because also in this case there is a prior that is somewhere and is retrieved immediately and put onto this percept, that we need a very high dimensional representation space. You cannot put all this into a Euclidean space, however complicated it is organized. The hypothesis is that such spaces can only be configured as dynamic spaces, because in dynamics you have a very, very high dimensionality. And the proposal is that recurrent networks actually have the property of providing such high dimensional coding spaces. Now, the hypothesis I would like to pursue and then discuss with you is that the evolution of two structures in the brain, the hippocampus and especially the neo-cortex, have introduced a novel computational strategy that capitalizes on the unique dynamics of recurrent networks. If you look closely, hippocampus and cerebral cortex are the only structures in the brain that have reciprocal excitatory recurrent connections. This is why they are susceptible to epilepsy. They are the only structures that can show epilepsy, but apparently the gain of having these structures compensated by a large amount the disadvantages, this danger of running off dynamics, running away dynamics. So, what are the dynamic properties of recurrent networks? I already said they provide an extremely high dimensional state space. Then they have another interesting property. They exhibit fading memory due to reverberation. If you tickle them, they will remember this for some while. And this permits superposition of information. It permits parallel search and fast matching with latent priors. I will give you a reason for that. It permits, because of this fading memory, to encode sequences, which is something that these deep learning networks have great difficulties to deal with. And these are properties that are exploited by still a small group of computational people, and they call it reservoir computing or liquid state machines. Here you see the names of those who pioneered this. So who is familiar with the concept of liquid computing? One. So it's worth going through it. By this metaphor, imagine a pond of water, still. You throw in a stone, you get a propagating wave pattern, so the water remembers for a while what has happened. You can then show that you can throw in a number of stones in a sequence, a big one here, a small one here, changing the energy and the size of these things and the places. These each will produce a wave pattern, and then there will be a very complex interference pattern resulting from this. And the theorem shows that if you just take 3 sensors in this pond and measure amplitude, phase, and wavelength of the oscillations that you see there, frequency, you can train a classifier to the sequence that has given rise to this pattern, and this classifier will perform 100%. And you can show that with this simple liquid you can discriminate a large number of different sequences and can train different classifiers. So this is used for language processing in analog systems in the moment. So they actually use a liquid. Now, one can implement some of the properties of the liquid with a recurrent network because it also has these abilities to create propagating waves. We talked about propagating waves yesterday. So I would like to propose an alternative view on the neocortex that really contrasts with the classical serial feed-forward strategies. I claim that the supragranular cortical layers can be considered as a delayed coupled recurrent network. Delay is important because it allows to play with phase space much more generously. Like also the waves in the water, they have a propagation delay. In the cortex, unlike in these simple networks that used for liquid computing, the nodes of the cortical network, they are feature selective. They are columns that respond to particular features selectively. And as I have already mentioned, most of them are damped oscillators. They can oscillate. A further claim is that the priors that I have been talking about are stored in the weight distributions of these recurrent connections. And they are abundant. This is a huge network, and the combinatorial complexity is quasi infinite. So weights be stored in the recurrent connections. It's not unrealistic, because this is how cortex look like. You have columns with particular feature preferences that can oscillate, and they are connected to these reciprocal collaterals, and there is a nearest neighbor scheme. So columns that are close together tend to talk together in a rather indiscriminate way. But as distance increases, connections become more and more selective, selectively coupling columns that have the same feature preferences, i.e. that code for features that have a high probability of occurring together in the outer world. So, now we provide you with evidence that these connections are actually adaptive. They learn about statistical contingencies. So let's see how far we get. There are a number of predictions one can derive. One is that the resting activity produced by these networks should have a very high dimensional dynamics, and it should reflect the superposition of all the priors, because they are latently there. They are not realized. It's like, for those who like quantum physics, it's like a superposition of wave functions that are not realized. But this is not quantum world here. The next prediction is that stimuli that match some of the priors of these predictions immediately induce a collapse into a low-dimensional substate of these network dynamics, like a nest of synchronized oscillations would be low-dimensional. And these substates, so goes the hypothesis, are easy to classify, because in such a high-dimensional space they will be nicely segregated, and they would actually be already the result of this matching operation. And they will be sparsely coded. But I won't talk about this. These result states, since they produce nicely coherent bouts of activity, they induce synaptic gain changes, which then are equivalent to an updating of priors, and also they are propagated forward much more easily than the dissociated activity that you see during resting state. So some first evidence. What we did is we did multi-site recording from visual cortex, many electrodes, presented stimulus sequences, and then tried with machine learning techniques to find in the vectors that were produced the information of the stimuli, see whether we could do it. Randomly selecting neurons, not caring for receptive fields and anything of that. Just select neurons. So here it goes. The bunch of neurons, 60 here, no 50 in that case, receptive fields mostly superimposed, presenting letters and numbers. And then sequences of them, an A, a B, and a C. Here are the raster plots of the responses. All these dots are spikes. We then convolved these point processes with a sawtooth function to get the continuous signal, which is easier for classifiers to digest, mimicking a little bit the EPSPs, smearing temporal relations to our disadvantage, but that's how we did it. And then train a simple linear classifier and change the weights of these input vectors to this classifier so that the classifier would, if there was an A at a particular moment in time, say there was an A. So once we had trained these classifiers, we looked as to the persistence of information in this network, real cortex now. Showing an A or a D, it took, interestingly enough, we could classify right from the beginning from the primary responses, but not so well, as you see. After a while, something has happened in the network that allowed us, after the stimuli had already been gone, to classify, or allowed the classifier to detect whether it was an A or a D, all the way up to 1 second. So the network memorized what it had seen. And this information could be retrieved from this high-dimensional vector. Oops. We also saw that information can be superimposed. we were able to find out, after the second stimulus had been shown and produced its responses, what the first stimulus was. Classification after having disturbed the network with the second stimulus is nearly as good as before. And this had already predicted now that we should be able, at a particular moment in time, from the vector that we get, to find out whether the first stimulus, what the first stimulus was, and what the second stimulus was. And this is a moment in time about here or here where this can be done, or here. So this means information can be superimposed in the network of different stimuli. And the network can even do an XOR operation, so find out what the first was, whether the first was an A or a B and the second was an A or B. This is a nonlinear operation. So the network did the nonlinear operation for us that a linear classifier could then profit from, because our classifiers were linear. So in summary, information is distributed across many neurons. We didn't look for receptive fields or any specificity, feature detecting stuff. There's fading memory, there's persistence of information across stimuli, there's superposition of information, and there's preservation of sequence order. So all this is compatible with computations in recurrent networks. Now there are further predictions one can derive. One is that the recurrent connections should undergo Hebbian modifications in order to store feature correlations of frequently occurring stimuli, imprint priors, required for the acquisition and updating of priors. So developmental studies show that this is indeed the case. And here's old stuff from our laboratory. This was before the time I got sidetracked by this oscillation stuff. That shows very clearly that these reciprocal connections that I'm talking about, they are really storing the statistics of the environment. What you do is you map functional domains. At these times was deoxyglucose. Nowadays one would do it with calcium imaging. And then functional domains is columns that are orientation selective or direction selective or ocular dominance columns. And then inject a tracer. At that time this was fluorescent beads. Nowadays it would be viral vectors or whatever. Doesn't really matter. And in a normal animal, you see there's a very high correspondence of connections between homogeneous functional domains. All these red and yellow dots are on the same functional domains, and they avoid other functional domains. This is an animal that had normal experience. If experience was withheld because they were raised in the dark, absolutely no systematic relation between those fibers. They grow out, they connect neurons, but they connect them randomly. There's a spatial decay function, but that's all. So these connections are selected, pruned, selectively consolidated according to functional criteria, and as we see here, according to a correlation rule. This is another experiment that's been done for a completely different purpose, where we exposed selectively kittens to a drum. Ernst, you may still remember, we did this stuff in Munich. And the kitten was always kept in the dark, but a few hours per day had this experience with these rotating stripes. So what happened is the kitten fixated these stripes, followed them, and then made a resetting saccade every now and then. The consequence of such an exposure is that cortical columns that code for vertical stripes, whose distance was in this case 10 degrees, is regularly spaced, they get co-activated all the time. Now these columns are connected through these horizontal connections that I've shown you in the diagram. So prediction, not at that time, it was an a posteriori thing. What we found is that 30% of the neurons in the supragranular cortex developed receptive fields with multiple centers. Not grid cells, but receptive fields with multiple centers. And one could map these multiple centers stationary with a bar flashed at different sites. The outside one here is a simple cell, a simple receptive field. This is a complex one, not much in between. Very robust responses obtained because of the Hebbian strengthening of connections between columns that were contingently active. Now, one can extrapolate from this. In the stereotyped case, it's simple, but if you are exposed to a complex environment, the same statistical evaluation will take place, and the contingencies, statistical contingencies of natural environments will be imprinted. Okay. Assuming that this is the case, and that these properties, okay, we will skip much, but doesn't matter. One expects improved classifiability of substates that are induced by familiar objects. So this is actually what happens in these experiments. One shows these patterns many times, and it turns out that late in the experiment, and you see again how long it takes before the network produces substates that are nicely classifiable. These are classification curves. The later, how long it takes to get these classifiable substates, first. And second, the red curves are taken from trials late in the experiment when the animal had already a lot of experience with these letters. And classification is much better. The reason for this is that in principal component space, the principal components of these vectors get segregated better late in the experiment. You see these three clusters here, than early in the experiment. And it also takes some time after stimulus presentation before these segregations occur. So the network self-organizes. Other prediction is that if these priors get stored in the network should replay them every now and then when it's just doing nothing, when it's resting. So we trained classifiers to reproduce the pixel values of patterns that we had shown. So if you present an F, we will see on the screen what the classifier has found. And then we use these classifiers and slide them over the spontaneous activity. And to our delight, we saw that every now and then, at irregular intervals, this bunch of neurons produces a vector that the classifier classifies as a 1 or an E, patterns that the animal had been seen. And to check, and something we couldn't properly interpret, and to check whether the classifier did what he should do, we presented a real C, and the classifier recognized it. So there is replay of these imprinted states. Then we switched to monkeys. Short of time, I will be fast. Prediction is that because monkeys have lived in a normal environment for quite some time, they should already have priors that correspond to natural images. Meaning that if we show them natural images, they should produce substates that are much more easily to classify than if we showed the same images, same energy in a scrambled way. So we do these chronic recordings with implanted microdrives, get these many neurons, and then show them natural images or phase scrambled images. Same energy, but no longer conforming to the statistics of real world. And we see this massive advantage in classifiability of the natural images over those that are phase scrambled. No changes in signal to noise ratio, no changes in discharge rate. Also, we see that when the incoming activity matches the priors well, like natural images, we have a remarkable ramp up of gamma oscillation power. So this is frequency, this is power, and this is gamma frequency after stimulus presentation. So you see for the natural images, there's a ramp up of gamma activity, and this goes with better classifiability, and is much stronger than in the case of scrambled images. And also stimulus identity, and this is perhaps for prosthetics important, can be decoded very nicely from the vector of local field potentials. So one doesn't have to have spikes. It can also be done by the matrix of field potentials. So I think I'm at the end of my time. Classification of scenes is possible, both from spike and LFP vectors. Classification is better for natural than scrambled scenes. Difference is not attributable to signal noise ratio and other changes, rate changes. The natural scenes induce stronger gamma, a stronger reduction in dimensionality. Does that mean? And so the proposal is that the match between natural scenes and stored priors leads to well-organized substates that are reflected by a gamma ramp-up and better classification. Now, I skip you a number of detailed evidences, and I think this is the summary. No. Yeah, let me summarize what I believe. I believe that indeed, with the advent of cortex, a new computational principle has been introduced that has been known to some extent to evolution for the pattern generators that are used for the generation of movement. But this option to produce complex patterns has not been used in order to be implemented in sensory systems. But once we have got this system, the computational abilities offered by these computations in high-dimensional dynamic space are so advantageous because they allow to, and now I come back to your T-shirt, allow to bind virtually everything with everything in the same data format, which of course is at the basis of abstraction, of symbolic coding, and all those wonderful things we can do. And my claim is this would not have been possible without cortex having been invented with this computation strategy, which I think we are just at the beginning of understanding. But my feeling is that one needs to pay more attention to the dynamic properties that the brain exhibits. It's probably not, as some people tend to say, the exhaust fumes of the working of the neuronal machinery. I think it is really functionally relevant. And with this credo, I would like to stop. Thank you.
Thank you so much. This was really a revelation for many of us. So, questions, so many questions. Yuri.
Wolf, I'm here. My recommended solution to the fast access of information is based on the log normal distribution of almost everything I know in the brain. So firing rates are log normally distributed. Faster firing neurons have stronger synaptic weights. They are better connected to each other. And they form what we can call a backbone of a pattern in the brain, which are non-plastic. Nevertheless, you can refer them, I refer them as the good enough brain that allows you to behave very fast in any situation. In contrast, I have to emphasize it's a continuum on the log normal distribution, but the other end is the low firing, low weight synapses and so on that are less connected, and they are represent an extraordinary amount of reserve for plasticity. How would you put this concept into your ideas?
Well, if I understood correctly, the low activity neurons, they would provide the reservoir for imprinting yet more and more new priors, modifying them, while the high frequency active ones would be a fixed backbone.
That would represent the priors, yes.
Yeah, and they could probably be, I guess the system can work in a simple feed-forward regime if there is no ambiguity. And it probably does so for in a number of situations. But as soon as it needs to resolve ambiguities, where it has to flexibly associate things with each other, it must rely on these more complex priors that cannot be built into individual receptive fields. Then you have to do the distributed coding. But for sure, the system must be in a state of close to criticality, and this may explain some of these log functions, straddling the state of chaos but not being there, in order to be able to descend rapidly and very freely into this huge number of local minima, because you don't have much time. I mean, if our system would have to work like the deep learning networks with these slow elements, it would go forever probably. So I think there's still something to be learned. It is probably not simply attractor dynamics, it's something else. It's the collapse of a high dimension system into low dimension substates. Yeah.
Sorry, before we go on with the questions, there were many questions here. I see you have a slide which you probably want to comment.
I put this up for Ajushi, because you mentioned so nicely yesterday, and this is something I tried out on the Dalai Lama recently, and it has to do with East-West, if you like. Just promoting the simple idea that as cognitive agents, perceptions, actions, and behavior, our properties emerge from neuronal interactions, there is a second step where these cognitive agents, if they start to interact in social dialogues, endowed with a theory of mind, reflect each other, they, of course, can produce culture and social realities. And the idea was that this should allow one to arrive at a naturalistic explanation of this mental dimension in which we participate, but it was an attempt to get away with ontological dualism. The Dalai Lama bought it. I don't know. And it reminded me of your talk yesterday, so this is why I put it in at the end. That's all.
Thank you so much. I think we should discuss this one also during the discussions. So Paul and Peter.
It seems to be moving from the sublime to the trivial, but could I ask, what is special about optimization in the gamma frequency band?
What is special about the gamma frequency band? In general, the higher the frequencies, the shorter the window of opportunity for communication. So if you want to be temporally precise, if you want to really exploit fully the SCTP rule, which is a picky rule because it is sensitive to millisecond offsets, then you are well advised to use precise temporal structure. So the gamma frequency would be, even ripples, they would be even more precise because things happen in the sub-millisecond range. Then there is this evidence for nesting oscillations of different frequencies, and that would be a possibility to express partial relations. So you encode one content in the cycle of a slow oscillation like a theta. Now if you have faster oscillations that you can nest into this rhythm, you can then establish partial relations between the items contained in the fast oscillation with this larger thing. That would be nice to produce hierarchical syntax, but there's no evidence so far. The faster the preciser, that's the message. And you cannot bridge too large distances with these fast oscillations and get them still in phase because of the conduction delay that gets into your way. So the very long-range communication is not happening in the gamma range.
It appears to me that Yuri is very keen to say something before Peter asks something.
Well, just to reflect this question, perhaps it's not what we have to ask what is so special, but perhaps what is so trivial about gamma oscillation. And what is so trivial about gamma oscillation is this is the time constant of the GABA A receptors. This is the time constant of the AMBA receptors. And gamma is nothing else but the tug of war between excitation and inhibition. So, but these are the two interesting issues. The most interesting one, of course, this is the duration or the time constant of the pyramidal cell membrane in the neocortex. So this is the time window within which upstream information can be integrated by the membrane of a downstream neuron. This is also the time window of spike time independent plasticity. So everything that is special about either plasticity or neuronal communication and transmission is expressed by gamma oscillations. Okay.
I just would add to that the NMDA receptor dynamic is much slower, and it's about half of the theta oscillation, and that's why theta exists on many systems because of the NMDA receptor activation and inactivation. But my question is somewhat related to Yuri's. Do you know evidence that within the same tissue space of supragranular neocortex, very small space, 100 micron or so, multiple pyramidal cell systems can oscillate and interconnect differentially? In cat and monkey visual cortex, the same response properties group in the same space and are segregated from different orientation, for example. But in rodent, they are intermixed, right next to each other cells with very different orientation selectivity. And if we think about higher order association cortices, where it's not so clear how many concepts they store, there may be within the same tissue space many different representations by differentially interconnected networks. But it's equally possible that simply the architecture excludes this, and that drives the multiplication of cortical areas, because they have to be dealing with the same information. So, for example, in the CA1 area, which we all think a rodent CA1 hippocampus is uniform, I showed 4 kinds of pyramidal cells, and it is now clear that within the same 50-100 micron, different pyramidal cell systems exist, and they have different response properties. So I'm very keen to learn of the supragranular, whether we know this and whether that's reflected in these assemblies oscillating, temporarily shifted. But of course, then they would interfere with each other, so we wouldn't see it in the LFP. I don't know the...
What I would take from it is that the different cortical areas staying in this dynamic world differ with respect to granularity, so the clustering of neurons with the like properties, and they differ with respect to time constants. There is more and more evidence that as you go forward and as you go more abstract, you slow down in rhythms. So that could be that the span or the amount of information that you can cluster into these units of objects of perception or objects of thought get bigger and bigger as you go forward. Now, how the pepper and salt cortex deals with the different feature domains, I would assume it does exactly the same thing. It selects the tangential connections as a function of the feature specificity of the neurons and their correlated firing. You don't have to have these big orientation domains in order to do that operation. So I think in resting state, neurons are fairly decorrelated in primary areas, except those very close together.
Apologies, I think you have some surprise to us. So we have a good 12 more minutes.
Why?
Can you just show what you wanted to show us? Wolf, it seems to me you have a surprise to us. Probably we should focus on this in the remaining 10 minutes.
Why do we have more time? Oh, it's not yet 12 o'clock.
Yes, so please go ahead, because this seems to be very intriguing.
No, this is just fun. So I was thinking that a condition that requires extensive definition of nested relations is conscious processing. Stanislas, if you talk about it, I won't. Will you? Okay. So, because he could as well do it. So there is some evidence, and Stanislas has pioneered this research talking about the workspace of consciousness, that there is large-scale temporal coherence of distributed neuronal activity in different frequency bands. It's mainly beta and gamma and theta, when subjects process stimuli consciously. So here's an example. Subjects were exposed to stimuli that could be either perceived or not. They were physically identical, but the masking procedure was such that 50% of the cases they wouldn't see what has been presented, were unable to report, which is the usual criterion for conscious processing. And what we saw, and Stanislas saw something very different, very similar, is that early on, after presentation of the stimuli, there is a brief epoch where large regions of the cortex engage in synchronous activity in the gamma or the beta frequency band. And this is not the case in the unconscious condition, even though physical conditions are the same. And then if one plots the networks that get activated, again, these were very simple detection tasks. In the conscious condition, one sees these widespread functional networks that are suddenly coordinated in time, and they are not appearing in the unconscious condition. So this reminded me of Sherrington. He had been cited already yesterday, who said pure conjunction in time without necessarily cerebral conjunction in space. He means one doesn't need these anatomical conversions in hierarchical structures, lies at the root of the solution of the problem of the unity of mind. So temporal coherence, defining what belongs together. So we seem to be quite good in finding the neuronal substrate of these cognitive functions, phenomenal awareness and so forth, but the question was, how about the spiritual dimension? And this was what the East-West thing suggested to me. Can we get around the ontological dualism, or what should we do? What is with our mental, spiritual, immaterial world in which we participate? And the proposal is simple. Once cognitive agents such as us that are endowed with the ability to enter dialogues of that kind, I know that you know that I feel and how I feel. So once you have this dialogue, you start to generate a new level of reality, which we call the social realities. Once we start to interact and image each other and reflect each other, this is what happens. And what is the nature of these realities? They are immaterial phenomena. They emerge from social interactions. They are absent in the pre-cultural world because those guys did not talk. They are not perceivable with natural senses. They could not be tuned to these dimensions because they were not around when the sensory systems evolved. And we commonly associate them with mental or spiritual or immaterial dimensions. And then there's a whole bunch of these social realities that we know of. They are very causally active because the content of a belief can produce us to make cathedrals or go to war or even kill ourselves. And so the proposal I would like to end with is that we attribute from these social realities a lot of the connotations that we associate with consciousness and the conscious self. And that by choosing this naturalistic account that we internalize in our self-model, what this cultural evolution has generated is different realities, that could perhaps help us to settle the epistemic dispute on consciousness, which is so difficult and so ill-defined. Also the dispute on mental causation, and in that virtue the mind-body problem. So if we consider these problematic connotations of consciousness as emergent properties of social reality that we self-attribute afterwards, then I think we get away with a lot of problems. And it also means we neuroscientists, we must not enter this field. This is a field that needs to encompass, in addition, the representatives of humanities. So as Archie talked about borders yesterday. This is a border we cannot explain from an individual brain those social connotations of consciousness. We need to reach out social neuroscience or even to the humanities. Okay, thank you for this 5 minutes.
We thank you, Phil, because this is exactly in the spirit of this conference.
Okay.
So we still have a few more minutes. So, Guy?
I'm sorry to come back to somewhat lower level brain-related question. So I have no difficulty at all to agree with you that binding is a very important principle. But I would submit that it's not the only principle. If we look at the evolution, what happens is that the sensory systems become more and more complex. There are more and more areas. And so what I think is that the reason why a human brain is so powerful is that we have increasingly higher levels of representation that are not present in simpler systems, and that then the binding problem becomes even more powerful because it's operating on much higher level representations than the one that you're studying in V1. And so to sort of put this in a very strong question, I would ask you, if the binding problem was so the way you portrayed it here, why did the evolution not choose for just a brain in which you have 5 primary cortices for each of the senses and nothing else, and then binding?
Well, your question comes down to the question, why didn't evolution opt for a very big retina? Because all the information is present in the retina. No, the reason is simply the same reason why one uses these liquids in order to do computations. There's a very important point in when you want to classify something. You have in a low-dimensional representational world, or scheme, or whatever, if you have many things that you need to classify, they would very quickly superimpose, and a classifier would have to be extremely complicated because it can no longer use a linear separatrix. It would have to produce a very, very complicated, and it's unclear how you would conceive of a classifier. What you can do, however, is you transform the low-dimensional stimulus world into a very high-dimensional space. And in my case, I did it in a dynamic space. You could also think about some relativistic space that is also very high-dimensional, but I don't think that the brain can do that. And then you segregate the representations in this huge dimensional space, and then you can classify very easily. And this is what liquid computing does for you. It transforms a low-dimensional stimulus space into a high-dimensional state space, does the classification, and then sparse codes the output. It's this up and down.
I can see your point, but then the critical experiment is to look at the high levels of the system and to see whether you have, what I think is that you have very complex receptive fields, not just in the spatial sense, but in the feature space, versus whether you have an amorph thing that is just doing your recurrent thing. So the critical experiment would be at the end and not at the beginning as you show in V1.
No, of course, one has to go. I think in the beginning I said I think we are at the very beginning of understanding. But what fits nicely is if you go up to the higher levels. Think about the paper of Churchland and Shenoy, for example, or the recent paper by Earl Miller. It gets tricky to talk about the receptive field because these neurons, they respond in a context. And if that context changes, these neurons change. And you would not be able to really say that this is a feature extractor or so. This is what you expect. They will have more complex response properties because these higher reservoirs, they have a different mapping rule. They have internalized different statistics because in that case, in the language system, they would evaluate statistics of vowels, of phonemes. I think there is no contradiction. What I don't like is to reduce our brain to a deep learning network.
No, I agree, and I think the example that you took in the prefrontal cortex, the recent work of Shenoy and Newsom is well taken. But on the other hand, some of the effects that you show with the shadow thing, I mean, that we have shown exists plainly in IT neurons. You know, we have lots of work on occlusion and things like that, and the effect is at the single cell level. So
yeah, of course. At the end, it must be at the cellular level. Of course, all this must have a neuronal underpinning. The question is, how do they get to these properties?
Yeah,
how do you get a neuron that has these particular properties? Can you do it simply by convergence and feed forward? I doubt it.
Agree. And then I agree, and then there's a level, but I think you reduce too much. But it is all that is happening in the sensory systems is far more complicated than we know because
I agree.
I've done some of this to show the gradient selective neurons that capture 3D shape and so on. So there are very high level representations in the visual system, and I think to operate on those high level increases the power of your priming system.
Yeah, I agree. We will find neurons for everything that we can do, obviously. The question is how can we find them, and then the second question is how are they coming to what they do? What is the mechanism behind it?
Sorry to intervene here. There are two more questions because we are really using our time. Two more short questions. One over there, one over here.
Yeah, just picking up if we can find neurons for everything we do, and if we can love, I'm interested in why you feel there must be a boundary between love, spirituality, and also the yin stuff and science. On the other side, you said, neuroscience should not go there. there have always been boundaries between spirituality and science. You know, I think Copernicus got into a lot of trouble with the Catholic Church by crossing that boundary. But these boundaries are very elastic and they change. So I'd like to understand why you think there should be a boundary and what that boundary is.
Well, I think it is a methodological boundary. What neuroscientists can do is find the neuronal underpinnings of the cognitive functions that specify human cognitive functions to a certain extent. Maybe even in the beginning of cultural evolution, we would still understand those things from a neuronal basis. But then, when the second layer of reality into which we have co-evolved developed, we need to change our description language. We cannot do this as we are used to talk about phenomenal awareness. This we can. These monkeys have it, and so we can analyze it. But these higher functions that are associated with the moving and the social realities, we have to look at these at this additional reference system.
So what you're saying is neuroscience can help us as a species get a better understanding, but not necessarily get a total picture.
Yeah.
So final question, short question, Peter.
And it's not very short, but it's very much related to this. So you showed or proposed that social interactions derive the spiritual domain and related to consciousness, and you used the word naturalistic explanation. I don't know why not biological explanation, why it has to be naturalistic. But the real question is if social interactions and communications contribute to that. Being in a technological university, where the communication of technology dramatically changed in our lifetime, and particularly in the last 20 years, completely changed. If you just walk out, everybody walks with a screen in front of them, interacting continuously. And the big news in the United Kingdom last week was that self-harm of females under 25 age increased exponentially since 2000 in the United Kingdom. Today, 1 in 4 females admits doing self-harm and needing treatment. 1 in 4 under 25. And some people draw a parallel with the social interaction and communication revolution. Some even proposes a causal relationship, but that, of course, remains to be established. So what is your prediction for the future of ourselves and what this meeting is about in light of the dramatic technological revolution?
I cannot predict the future. I would think that our self-model would change.
Would you speculate?
I think what we can say for sure is that our self-model, models that we attribute to ourselves, they will change under the light of scientific progress as it has done already in the past, and technological progress will probably also contribute to it. In which direction it will change? No, evolution is not predictable. It's non-linear.
So actually, this is the point when we should continue the discussion and not stop it. So I really feel very much embarrassed that we should, for practical reasons, stop the discussion now and announce a lunch break, and we convene at 1 o'clock. But before we go out of the room, let me thank again Wolf for this fantastic speech and the discussions which we should continue, definitely.